Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss fatty acid breakdown in peroxisomes. In the previous videos, we discussed various breakdown pathways, catabolic pathways that predominate during starvation states that include glycogen breakdown, triglyceride breakdown, the beta-oxidation or the breakdown of various kinds of fatty acids, including saturated fatty acids, monounsaturated and polyunsaturated fatty acids, as well as fatty acids that had off-number carbon atoms. Then we also discussed another type of oxidation besides beta-oxidation, and that was omega-oxidation, and this omega-oxidation led to dicarboxylic acids. Okay, so this leads to the formation of dicarboxylic fatty acids. In today's video, we shall look at lipid metabolism in peroxisomes. Now, peroxisomes are organelles that are rich in tissues that have active lipid metabolism, and these include tissues such as liver, adipose tissue, cells in the brain, as well as intestinal mucosa. Peroxisomes are organelles typically less than 1 micrometer in size, and they have a membrane that is a bilayer membrane. Since peroxisomes actively break down lipids, and we'll look at this in a few seconds, the drugs fibrates, which help reduce lipids in the body, actually increase the number of peroxisomes. And that increased number of peroxisomes leads to increased lipid breakdown that helps reduce the amount of lipids in the body. In a previous video, we talked about how peroxisomes are involved in the synthesis of ether lipids, for example, plasmalogens. We discussed how peroxisomes, as well as the smooth endoplasmic reticulum, are involved in the synthesis of plasmalogens. In today's video, we shall look at another type of oxidation called alpha-oxidation. And this alpha-oxidation is necessary for branched-chain fatty acids. So for example, you can see there's a branch here at position 1, 2, 3. And because of this branch, this fatty acid cannot undergo beta-oxidation. Therefore, it first has to undergo alpha-oxidation, and after alpha-oxidation, it will form a product that can then undergo beta-oxidation. So we'll look at two examples of branched-chain fatty acid. One is phytol, which is converted in the smooth endoplasmic reticulum into phytanic acid, and the second is phytanic acid. And we'll see this phytanic acid will be taken into the peroxisome, undergo alpha-oxidation that we'll talk about, and then phytanic acid is converted into pristanic acid, which then undergoes beta-oxidation in the peroxisome. We'll also discuss the beta-oxidation that happens in the peroxisomes for not only pristanic acid, which comes from the branched-chain fatty acids, but also for very long-chain fatty acids, where the N number is greater than 21. We'll also discuss beta-oxidation for long-chain dicarboxylic acid fatty acids. So there's a carboxyl group here, and there's a carboxyl group at the other end. These dicarboxylic acids come from omega-oxidation that we discussed in previous videos. And then we'll also discuss the bile acid intermediates which are formed from cholesterol, such as trihydroxycholestonic acid, which is shown here with 3 hydroxyl groups, or the di form with only 2 hydroxyl groups, THCA or DHCA. Let's begin with the alpha oxidation for branched chain fatty acids. Phytanic acid is a branched chain fatty acid, and as mentioned before, there is a branch at position 3, and because of this branch, this fatty acid cannot be broken down by beta-oxidation. It therefore undergoes alpha-oxidation, and in the first step, phytanic acid is converted into phytanyl-CoA on the surface of the peroxisome, and this is catalyzed by the enzyme acyl-CoA synthetase using ATP and attaching coenzyme A to the fatty acid, thereby activating phytanic acid. The phytanyl-CoA that is formed on the cytoplasmic side is then transported into the peroxisome, and this transport is carried out by a transport protein called PMP70, which belongs to the ABCD3 family of transport proteins. As a healthcare professional, it is important to also know the names of some of these transport proteins, because genetic diseases may affect not only enzymes but also transport proteins. So PMP is peroxisomal membrane protein, PMP, while the ABCD here stands for, the ABC stands for ATP binding cassette transporters, and D here stands for subfamily D. Once the phytanyl-CoA is brought into the peroxisome, the alpha carbon, this alpha carbon is oxidized using oxygen and making a hydroxyl group at the alpha carbon. This is carried out by the enzyme phytanyl-CoA hydroxylase, and this requires the transport of alpha-ketoglutarate, which comes from the mitochondria, because alpha-ketoglutarate is required for this reaction. Carbon dioxide is formed, succinate is also formed, which is also transported out of the peroxisome through this channel in the peroxisome membrane called the PXMP2 channel. And we also form 2-hydroxyphytanyl-CoA. So the alpha carbon has a OH attached to it and therefore is oxidized. So that's why we call this alpha oxidation. In the next step, the bond between carbon 1 and 2 is broken, and this part comes out as formyl-CoA, while the rest of the molecule is pristanyl. The OH is converted into an aldehyde. The formyl-CoA eventually breaks down by itself into formic acid and coenzyme A, and this reaction is catalyzed by a lyase. The pristanyl is then oxidized by NAD. The aldehyde group gets converted into a carboxyl, forming pristanic acid. This reaction is probably catalyzed by some sort of an aldehyde dehydrogenase, although we are not quite certain about exactly what enzyme is involved here. So through these alpha oxidation reactions, the methyl group, which was at position 3, is now at position 2 in pristanic acid. Pristanic acid now can undergo beta-oxidation. The pristanic acid is transported out into the cytoplasm, And there, this enzyme that was involved here will convert the pristanic acid into pristanyl-CoA, which is then transported back into the peroxisome, and in the peroxisome it will undergo beta-oxidation, which we'll talk about in a few minutes after we look at the metabolism of phytol. Now, besides phytanic acid, which we get from our diet, we also get phytol in our diet, and this is particularly rich in the skin of nuts. So let's look at the metabolism of phytol in the endoplasmic reticulum. How does this branch chain fatty acid is metabolized, how it is metabolized in the ER. So in the first reaction, the enzyme alcohol dehydrogenase, using NAD as an oxidizing agent, oxidizes the alcohol into an aldehyde. Phytol becomes phytanol. In the next step, once again, NAD is the oxidizing agent. The enzyme fatty aldehyde dehydrogenase oxidizes this aldehyde into carboxylic acid, converting phytanol into phytanic acid. In the next step, the carboxyl group is activated into a coenzyme A. Phytanic acid becomes phytanyl-CoA using the enzyme acyl-CoA synthetase and ATP energy as well as coenzyme A. Then in the last step, this phytanyl-CoA, the double bond here in phytanyl-CoA, is reduced by the enzyme enoyl-CoA reductase using NADPH as the reducing agent. So we get a single bond here and we get the product phytanoyl-CoA. So phytanoyl-CoA basically is what we have here, and now the rest of the metabolism is similar to what we talked about, the alpha-oxidation. This phytanoyl-CoA will eventually be converted to pristanic acid, and this pristanic acid will undergo beta-oxidation. So eventually phytol and phytanic acid will be converted to pristanic acid, and that pristanic acid will undergo beta-oxidation that we'll talk about next. So now let's look at beta-oxidation that occurs in the peroxisomes. So we'll focus on very long-chain fatty acids, then we have this long-chain dicarboxylic acids, then we have the bile acid intermediates such as THCA or DHCA, and then we'll look also at pristanic acid, which is formed from phytol as well as phytanic acid. So all of these will undergo beta-oxidation. In a previous video, we talked about beta-oxidation in mitochondria. Now let's look at beta-oxidation in peroxisomes. So the fatty acid is transported into the peroxisome. For the peroxisome, carnitine is not needed to bring the fatty acid into the peroxisome. Carnitine was needed for mitochondria. Remember, we need this carnitine transport system to bring the fatty acid from the cytoplasm into the mitochondrial matrix, but for peroxisome this is not necessary. Now in the mitochondria, the first step was an oxidation using FAD as the oxidizing agent. This was the enzyme acyl-CoA dehydrogenase, and the FADH2 that was formed, those electrons got transported to the electron transport chain by these two proteins. In the peroxisome, we once again have FAD involved, but the electrons are transferred to oxygen instead of the electron transport chain. And this oxygen becomes hydrogen peroxide. And this hydrogen peroxide can form dangerous free radicals, so it has to be broken in case they are present in excess. So peroxisomes are rich in this enzyme catalase that can break down excess hydrogen peroxide into safe chemicals like water and oxygen. So this oxidation between carbon 2 and 3, just like what we saw in beta-oxidation in mitochondria, this oxidation forms a double bond, and the enzyme here in peroxisome is called acyl-CoA oxidase 1. Acyl-CoA oxidase 1 is involved in very long chain fatty acids as well as dicarboxylic fatty acids. For pristanic acid as well as the bile acid intermediates DHCA and THCA, there is another enzyme called ACOX2. Okay, so instead of ACOX1, the ACOX2 is involved in this reaction for pristanic acid and for the bile acid intermediates. There's also another enzyme that is necessary for pristanic acid and the bile acid intermediates, and this enzyme is racemase, and this enzyme converts the 2R isomer of these chemicals into the 2S isomer, which is necessary for their metabolism. So this oxidation here leads to the formation of trans-2-enyl-CoA, very similar to the mitochondria. In beta-oxidation in the mitochondria, the next step, remember, is addition of water, what we call hydration. That was done by this enzyme in the mitochondria, which is part of the MTP, this hydratase. In case of peroxisome, the same hydration is carried out. Basically, add water across a double bond to form a hydroxyl here. But this is carried out by the enzyme D-bifunctional protein for very long chain fatty acids, as well as for pristanic acid and THCA. However, for dicarboxylic acids, the enzyme that is used for this step is L-bifunctional protein. In the next step, the hydroxyl group at the beta carbon is oxidized by NAD to a carbonyl. So since the beta carbon is oxidized, that's why this is also called beta-oxidation, just like what we saw in the mitochondria. In the mitochondria, the enzyme that carried out this reaction was the 3-hydroxyl acyl-CoA dehydrogenase that was part of the MTP protein. In case of peroxisomes, this reaction, similar to the previous reaction, is catalyzed by D-bifunctional proteins for very long-chain fatty acids, as well as for pristanic acid and the bile intermediates THCA and DHCA. However, for the dicarboxylic acids, there is another protein that catalyzes this reaction, and that's called the L-bifunctional protein. So this oxidation converts the 3-hydroxyl acyl-CoA into 3-keto-acyl-CoA. In the last reaction, the bond between the alpha and the beta is broken, and that releases this part out as acetyl-CoA, and the remaining part is basically your fatty acid shortened by 2 carbons. In the mitochondria, this reaction was catalyzed by 3-keto-acyl-CoA thiolase, breaking of this bond between the alpha and the beta carbon. In case of peroxisomes, for very long chain fatty acids, we have 2 enzymes that can do this. One is acetyl-CoA acyl transferase 1, also called peroxisomal 3-keto-acyl-CoA thiolase, or ACAA1 or PTH1. The second protein that is known to do this reaction, catalyze this reaction in peroxisomes, is called steroid carrier protein X, or SCPX. So unlike the very long chain fatty acids which have two enzymes that can catalyze this breaking of this bond between the alpha and the beta. In case of dicarboxylic acids or pristanic acid or the bile acid intermediates, only this protein, the sterol carrier protein, can catalyze this last reaction. In case of very long chain fatty acids, then this fatty acyl-CoA that is formed here will go back to the cycle and undergo another round of beta-oxidation, and this keeps on happening just like in mitochondria. But in case of peroxisomes, this beta-oxidation continues happening until the very long chain becomes into either a long chain or a medium chain fatty acid. And once we get into the long medium chain range of fatty acid, then that fatty acid is transported out by carnitine octanyl transferase. So in this case, for the transport of the fatty acid out of the peroxisome, carnitine is required. And then eventually this long or this medium chain fatty acid will then enter the mitochondria and be completely broken down by beta-oxidation in the mitochondria into acetyl-CoA. The acetyl-CoA that is formed is also converted to acetyl-carnitine. So once again, we require carnitine, and this is then transported out of the peroxisome by carnitine acyl carnitine translocase. And the acetyl-carnitine then is converted to acetyl-CoA, and this can be used by the cell for various purposes. So although carnitine was not required to bring the very long chain fatty acid into the peroxisome, we see here that carnitine is required to transport the fatty acid as well as the acetyl-CoA out of the peroxisome. So just to summarize, the very long chain fatty acyl-CoA will go through beta-oxidation in the peroxisomes, eventually being converted into either long chain or medium chain fatty acid, and this is then transported to the mitochondria, where it undergoes further beta-oxidation to break down the fatty acid. In case of the dicarboxylic acids that are formed from omega-oxidation, they go through beta-oxidation in the peroxisome, forming acetyl-CoA and the dicarboxylic acid shortened by 2 carbons, and this then go back into the cycle, and this keeps on happening, and it's believed that this keeps on happening until it is completely broken down into the dicarboxylic acid called succinyl-CoA, and this succinyl-CoA can then be used for the Krebs cycle. So this could be a way to make succinyl-CoA for the Krebs cycle. For the bile salts, the side chain undergoes beta-oxidation, and then it is converted into the bile salts, toro or glycoquinodeoxycholate, as well as toro or glycocholate, and these bile salts are then transported out into the cytoplasm from the peroxisomes and eventually come out into the bile, and this is predominantly carried out in the liver. Pristanic acid is taken into the peroxisome, goes through beta-oxidation until it's converted into a shorter fatty acid, dimethyl-nonanyl-CoA, which then goes through beta-oxidation in the mitochondria. So let's look at this part here a little bit more. So peroxisomal beta-oxidation of pristanyl-CoA. Okay, so let's look at these steps in a little bit more detail. What happens to pristanyl-CoA when it undergoes beta-oxidation in the peroxisome? Remember, pristanic acid is converted to pristanyl-CoA. So basically, pristanyl-CoA comes from either phytanic acid or from phytol. Both phytanic acid and phytol, you can see, are branched chain fatty acids. So now let's look at this part here. What happens to pristanyl-CoA when it goes to beta-oxidation in the peroxisome? So in the first cycle of beta-oxidation, once again, we break between the alpha and the beta carbon. This part here will come out as propionyl-CoA. So wherever this branch is, you can see this is going to come out as propionyl-CoA. And this propionyl-CoA is then converted to propionyl carnitine to be transported out of the peroxisome. In the next cycle, once again, we break the bond between the alpha and beta in beta-oxidation. And now we have this part here, and this part comes out as acetyl-CoA. Okay, so this acetyl-CoA is converted to acetyl-carnitine, which is then transported out of the peroxisome. And in the third cycle, once again, we break between alpha and beta, and this is again a branch area. So we get propionyl-CoA, which is converted to propionyl carnitine, and that is transported out of the peroxisome. Now this remaining part here will be 4,8-dimethyl-nonanyl-CoA. If we count the carbons here, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's nona. So this is nonanyl-CoA. And then at carbons 1, 2, 3, 4, and 5, 6, 7, 8. So at carbons 8 and 4, there is a methyl group. So this is 4,8-dimethyl-nonanyl-CoA. And this fatty acid is transported into the mitochondria, and there it is broken down into acetyl-CoA as well as propionyl-CoA. Just for completion, let's see how this dimethyl-nonanyl-CoA will be broken by beta-oxidation in the mitochondria. So from the peroxisome, this will be transported into the mitochondria. And as before, mitochondria, to transport the fatty acid into the mitochondrial matrix, we need carnitine. So carnitine is required. And so what we get is 4,8-dimethyl-nonanyl-CoA transported into the matrix by the carnitine transport system. And once it's in the matrix, it undergoes beta-oxidation. So the first cycle releases this as acetyl-CoA. Then the second cycle, once again, we have a branch here that is going to release this part here as propionyl-CoA. And then the next cycle will release this part here as acetyl-CoA, while the remaining fragment here will be isobutyl-CoA. Okay, 1, 2, 3, 4. 1, 2, 3, 4 carbons. So this is isobutyl-CoA. So the breakdown of pristanyl-CoA, pristanic acid, not only gives us acetyl-CoA and propenyl-CoA in the peroxisomes, which are then transported out, but in the mitochondria we also get acetyl-CoA, propenyl-CoA, as well as isobutyl-CoA. So this is how we break down branched-chain fatty acids by alpha-oxidation, and once they are broken down by alpha-oxidation, they are converted into products such as pristanic acid that can then undergo beta-oxidation, and then the other fatty acids, including pristanic acid, can undergo beta-oxidation, and these include very long-chain fatty acids, the long-chain dicarboxylic fatty acids, as well as the bile acid intermediates, especially this chain here, which you can see is looks like a fatty acid. They all go through beta-oxidation and are broken down either in the peroxisomes as simpler products, acetyl-CoA, propenyl-CoA, or are sent to the mitochondria, where they are broken down into simpler substances such as acetyl-CoA, propenyl-CoA, and isobutyl-CoA. So with this discussion, we see that fatty acid oxidation occurs not only in the mitochondria, but is also a very important process that happens in peroxisomes. So that is all for this video. In the next video, we shall leave early starvation and will now talk about the metabolic pathways that predominate during late starvation. So until the next video, thank you very much for watching. Goodbye and have a happy and a healthy day.